Diptanshu has been working with me for 3 terms. In the first term, he worked with a group that did spatiotemporal entanglement in quantum physics. He decided that he wanted to move into quantum computing, and he has done 2 terms of work in using quantum computing to improving computing speeds, and he's going to explain to you today how he is using this methodology for machine learning using neural networks. So Diptanshu, take it away.
Okay, thank you so much. So hello everyone, I am Diptanshu Sept and welcome to my presentation on quantum convolutional neural networks via variational quantum circuits for efficient image recognition or image classification. So our team consisted of Max 3, Adrian Cao, Abik Das, and Amit Baira, and we are the quantum computing group from Dr. McMahon. Let's get started. Yeah, so machine learning, as IBM puts it, is a branch of artificial intelligence and computer science which focuses on the use of data and algorithms to imitate a way that humans learn, so gradually improving its accuracy. As the world has seen so far, machine learning is extremely useful to solve many applications to which humans cannot derive a mathematical model that takes care of both the obvious and hidden patterns in the data. However, machine learning does have some limitations, especially when learning from large amounts of data or very complex data sets with many features. Because machine learning fundamentally relies on probabilities, some computer scientists have turned to quantum computing to increase both the accuracy and efficiency of machine learning models, thus introducing the field of quantum machine learning, or QML for short. So QML is a multidisciplinary field involving both machine learning algorithms and quantum computing concepts. It has numerous possibilities and can outperform classical machine learning algorithms due to its phenomenal computational speedup or reduced computational complexity, however way you look at it, and vastly increased capacity for computation. To test the quantum advantage in the noisy intermediate scale quantum era, which is basically like, we don't have a perfect quantum machines right now because there are a lot of errors that happen with qubits themselves. So what John Preskill, a professor in Caltech, has named the era it's the NISQ era, which is basically the era of imperfect quantum machines, essentially. And so we were building on that, and we were implementing a quantum convolutional neural network in that environment using TensorFlow Quantum. So we hypothesized that our QCNN would have a higher accuracy than a classical convolutional neural network, and our QCNN structure or architecture parallels the classical structure of a CNN, except that it is built in the quantum domain. And we have taken the quantum classical hybrid approach, basically utilizing both quantum and classical computers to achieve, to basically do both sides of machine learning, like optimization or training, to whichever the
你干嘛要定在周四呢？
一周2次那样，周2、周4。
The aspect is good in. So the subject of our research is quantum machine learning, as you know, but to understand QML, we must first talk about its predecessor, our machine learning, that is a little more in depth. So machine learning is performed on classical bits and mimics neural networks of the brain. A machine learning model consists of several layers which can process data and produce an output. And these layers have a set of weights or neurons associated with them, which control how the model processes the data. To train a model, the training data goes into an untrained model, of course, which then does an initial classification. these values are compared to the actual values, yielding the loss or how far off the model is, and which can then be used to calculate the MSE or the error function, basically. MSE stands for mean square error. So the model learns from its mistakes, which are quantified by the MSE, and the errors are then back propagated through the model, and using an optimization algorithm, such as adaptive moment estimation algorithm, or ADAM for short, the parameters or weights are adjusted so the model can be more accurate. In the context of quantum ML, which is relatively newer, the model will have layers of quantum circuits instead of classical layers, meaning that instead of just ones and zeros, we'd have quantum states, which can be anything between or a combination of 0 and 1. So one way to sort of, I guess, think about it is a bit can either represent 1 or 0 at any given point of time. But then a qubit can represent 0, anything between 0 and 1. Essentially, it's basically like a probability distribution between 0 and 1 states. So I could have the qubit could be in a state of 40% 0 and 60% 1 at the same time, which a bit cannot do. And so this has like long, far-reaching consequences, which may differentiate actually quantum computing from classical computing. And it really presents quantum computing as a solution to certain kinds of problems, not all problems, because there are other kinds of like computation heavy problems that classical computers are much better at doing than a quantum computer. So anyway, the goal of the research was to create a quantum convolutional neural network for image classification to compare with a classical convolutional neural network. And research literature states that a QCNN should be more efficient, and we try to test that out specifically for the identification of handwritten digits. So we have several methods for improving the performance of CNNs, and we augmented the pre-existing framework of variational quantum circuits, which are commonly used in speech recognition and are proven to be effective and applied in this algorithm for efficient image recognition. Now, our QCNN structure is the intersection between the structures of traditional convolutional neural networks and variational quantum circuits. A classical to quantum encoder is added to make inputting classical data into the quantum domain. So because bits cannot just be transferred into qubits, so that we need an encoder program. Then we have a set of cluster state quantum circuits to entangle and superpose all these qubits, and basically the initial quantum states known as the inputs. And we use an ingenious encoding algorithm for our encoder. And this algorithm basically transforms a grayscale image of dimensions 28 by 28 pixels into a two-dimensional array of zeros and ones at the same size. Now, we did this because our images were they were grayscale, but they're only grayscale. So we could round all the pixels. They would have a value from 0 to 255, about like telling between white and black or anything in between. And we set a threshold. If you're greater than, I believe, 160, then you're one color. And if you're not, then you'd the other color. So we've made a grayscale image a total black and white image. So that's what that encoding does. Now, these values then get passed into the cluster state quantum circuit, and which applies Hadamard gates to each qubit and the controlled NOT gates on each pair of consecutive qubits. So 1 and 2, 2 and 3, 2 and 4, 4 and 1, and to entangle the states. Next, we have the Pauli-Y rotational gates are added to implement a phase shift. So the qubits spin about the y-axis instead of the usual x-axis. After this follows a series of alternating quantum convolution and quantum pooling layers, QCLs and QPLs. And in the QCLs, there are unitary and controlled unitary gates to perform quantum convolution with trainable parameters. Basically, instead of having a fixed function within each gate, we have parameters, like pre-parameters, basically. And our goal is to train these parameters when we do the QCNN training and testing phase. So these parameters specifically are theta, phi, and lambda, which correspond to the rotations about the x, y, and z axes. And this differs from the unitary circuits found in VQCs, which have rotational angles chosen from a uniformly random distribution. So oftentimes you will see that people who implement variational quantum circuits, they choose to do random parameters, and it sometimes works, it sometimes doesn't work. It really depends on what the number or what the angle that is chosen and how the data set is. So it's not the best form of deterministic programming. And in the QPLs, we have that is pooling layers. Pooling is implemented to pull down n qubits to n by 2 qubits, so 4 to 1. And in the last QPL, we also measured all the qubits to yield the probability result of the model. Then the mean square error, or MSE, is calculated, and the VQC rotation angles are optimized using the atom optimizer, which is actually done on a classical computer, before repeating the process several times to acquire the best rotational angles. So at the very end, we decode the output in order to recognize which of the handwritten digits the input originally was. So as you can see on the log diagram on the right corner, we start with the inputs. We actually take 4 qubits in, and that 4 is a specific number because we had a 28 by 28 pixel image, and we wanted to comb through the image using a 2 by 2 pixel window. Thus we get 4 pixels, and that's when we have 4 qubits. So we start with 4, then we do unitaries for U and controlled unitaries or CU, and Z is just a phase flip. And we basically iterate the process 2 times, and we get the output, which is again measured, and then we transfer it back into classical domain. Okay. Now we'll be digging down into like how the encoding, I did mention it earlier, but we'll be digging more down. So the first step in the QCNN process is really encoding classical data into quantum data. This step entails converting 28 by 28 pixel images from the NMIST dataset, which contains grayscale values, or a single byte that stores a value from 0 to 255 to values that can be stored in qubits, or values from 0 to 1. We employed a novel method of encoding that involves simply turning an image into a list of RGB values, and then we using this list to create a new list of binary values representing either 0 or 1. So the binary value for a certain pixel in the image is decided based on a threshold, which every value greater than or equal to that threshold becomes 1, and every value less than the threshold becomes a 0. This threshold was decided through testing, repeated testing, that is, mainly with numbers 9, 6, and 0, as pictured on the right corner here. And because the curves would be easily lost in 2 ways. Either the thick lines combined with the high threshold would cause simple circles without holes, or not thin lines combined with a low threshold would cause gaps within the curve. So we didn't want either of those. And so after the process of applying the threshold to each value, an array of encoded values is ready to be inputted into the qubits for convolutional pooling. So now basically the heart of this entire algorithm, QCNN, the variational quantum circuits. So they're also known as parametric quantum circuits, and they're basically algorithms that that depend on free trainable parameters. So similar to any quantum circuit, the variational quantum circuit has three main components. There's the preparation of an initial state, a quantum circuit with a set of free variables, often denoted by the Greek letter theta, and a measurement of the qubit states in the circuit. So quite similar to machine learning, the VQC's rotation parameters need to be also trained by an optimization algorithm. So because classical computers are much better suited than quantum computers at optimization, VQCs rely on classical hardware to find the best candidates for the parameters theta via an iterative process. And an example of recent use of a VQC is the variational quantum classifier, which is also VQC, which uses two variational circuits. The first circuit associates the gate parameters with fixed data inputs, while the second circuit depends on free, the trainable ones. And together with the final measurement, this setup can be interpreted as a machine learning model of the simplistic kind, but it is one. And so we kind of use the same idea in our QCNN. And we also use a series of VQCs in order to reduce the chances of biasing the model in any way. So just to touch upon the convolutional neural network methodology, to clarify what is QCNN, essentially that question. So let's look at CNN quickly. So CNNs are a variation of artificial neural networks, or ANNs, and are most helpful in applications like image recognition. A CNN avoids overfitting by not making connections with all the neurons, unlike the ANN. And this is very important because a lot of times when we have ANN, they're very good in the training data. They achieve maybe 99% accuracy, but when you're testing, the accuracy of course drops because you overfitted, you learned too much kind of thing, and you learned so narrowly that you only know what's in the data set and you can't really infer anything else. So CNNs have three types of layers. So they have the convolutional layer, the pooling layer, and a fully connected layer. So the convolutional layer enables the model to perform efficient feature extraction from the data, one of its main highlights. And in the deep CNNs, we would have multiple convolutional layers, and they'll be stacked together to recognize more complex patterns. But there's also like pooling, which is used to reduce the dimensionality and size of the model and complexity. Basically, it performs a lossless compression of information or learning, depends how you look at it. So we did complete the internal architecture of our QCNN, and including the quantum convolutional layers and pooling layers. So we're still actually training our model in the sense for images, for handwritten images. So our current results are not available. We'll be working on that a little bit in this semester. And our next action item is to really make that work. And afterwards, we'll train and test. And then based on the existing literature research and our analysis of other QCNNs, because there are like a couple others that have been built, we theorize that our success rate will be somewhere in the 70s, 80s, could be that high. Of course, you may be wondering, well, CNNs can do much better. They can do 98% accuracy, as I've seen in one article. The catch here that I want to really highlight is that quantum convolutional neural network, any quantum neural network or any quantum program for it in that itself, it's going to underperform classical computers or classical algorithms for small data sets or data sets with less features, because we're not able to harness all the power of QCNN. So it may take longer to train, it may get confused and get stuck in a local gradient. It could be all kinds of stuff. And there's actually complexity of this, like the big O notation for any quantum algorithm, especially QCNN, is I believe long. base 10 to the base log of n base 10 versus the classical networks are n square of the order of n square. So in the beginning, the classical neural network would be less expensive than the quantum. But over time, as we get into higher dimensional data, we have the quantum overpowering the classical. So anyway, we did test our model on TensorFlow state excitations data set, which is a more quantum mechanical thing. It's a quantum mechanical problem. And we did it to check the functionality of our QCNN, and we did achieve around 98% accuracy, which is very promising in the sense that maybe if we have much more data in the future, or we try to solve a different kind of quantum mechanical problem, we'll probably be able to get a higher accuracy. So we customly constructed QCNN, and we had a BTC, and we also trained the parameters with that state excitations data set. And during our research, we actually had several challenges, such as information loss during the encoding phase. The information loss during encoding was because the information in each bit was rounded based on a threshold, but we kind of figured that out. I think we also had trouble on really choosing which gate to use in the QCNN layer. So there are many gates in quantum. There's logical gates, there are phase gates, there are all kinds of gates. And basically we're in that phase that classical computers were 30 years ago, in the sense we have to hand draw the circuits which are actually going to be used in the algorithm. So many established quantum algorithms, they basically create an oracle of basically a function of multiple gates, and they choose the gates, and we weren't getting that recipe. So we had to like comb through a lot of GitHub files and a lot of people's research, read a lot of papers to find really like, why am I going to put some rotation gates in some order, or why or how am I going to do phase flips? Like, why should I do that? Why should I really put that there? Right. So that was one thing we struggled with a lot, and we were able to get through it. And we have like different versions. We have like 6 different versions of our convolutional layers, just using different kinds of gates in different orders. So yeah, that was one of our solutions, to try out multiple quantum circuits. So yeah, in the sense this is our references, but so in terms of acknowledgements, I would first and foremost, I would like to thank our research advisor, Dr. Larry McMahon, for all his feedback and encouragement. I would also like to thank the ASDRP for providing us with the opportunity to conduct this research. And lastly, I would like to personally thank my team members for their amazing hard work, patience, and perseverance through this project. Thank you.
Thank you, Diptanshu. I thought you did an excellent job presenting this to us, and I will hand over the mic to our admin now.
I think we have questions. Sahar has a question.
Questions, yeah, question.
Yes, Diptana, awesome job. It's a very interesting topic when you touch artificial neural network. So did you try to use the approach of decomposition approach? I mean, because, so are you familiar with decomposition approach? No. Okay, so let me give you an example. So one of the easiest way to categorize number is decomposition. So so there are 2 features only in that machine learning algorithm. One is the intensity, and the other one, if I'm not wrong, is symmetry. For example, number 5, when you classify, has higher, when you voxel, divide it into the voxel, basically you go from multi-class to binary. So what will happen, you do voxel analysis, right? for that decomposition approach, the intensity for number 5 will classify 5 into the intensity feature because, of course, it's occupy more voxels. On the other hand, you have number 1, which goes under the symmetry. So when the classifier try to classify based on symmetry or intensity, like it can see 5 and classify it as 5 and 1 as 1. Okay, so that's a pixel-based analysis. So what will happen? So there's one of the approach for that is PLA, which is called, I think it's called perceptron learning algorithm. So I would recommend you check for that because that's the easiest way, like before you start. So your work is pretty advanced, but just like as in starting like a machine learning algorithm, like it is required that they start with a linear regression, which you are familiar with that, right? So PLA will help you to start doing voxel analysis using the linear regression and using the classification to classify the numbers. So I would say, I mean, like, as in like, just a recommendation, just give it a try since it is just like a primary step towards that, and see if there's like there's PLA and there's Pocket. So just like you want to see, you want to test your algorithm versus that. So because it's a very standardized one, and like people who enter to machine learning, they start doing the number and alphabet recognition. So they start with PLA and coding that. So basically that's a first step. So just like, and they give a 99.5 or 6% accuracy for that, you know, because that's the simplest algorithm with less feature. So just like, I mean, your work is pretty awesome, it's pretty advanced. Just try to, you know, find this little algorithm and just implement it to just make it prove that you have already, you can also cross that error, like you can even cross 99.5 to get that accuracy done. But I think your work is wonderful. So it's called PLA. I think it's called perceptron learning algorithm, if I'm not wrong, in PLA and Pocket. So that's one of them. And then the other one is called decomposition approach, which the 2 feature just for intensity and for symmetry. So I would like to hear once you code it and see how, you know, your ML works on that.
Sure, thank you for that. Oh,
You're welcome. You're welcome. I definitely look forward for that. Good job, well done.
Thank you. Any other questions?
Other questions?
One of my teammates, he messaged me, how about a quantum perceptron learning algorithm? So QPLA, maybe, I don't know. No other questions? Nothing on the methodology for QCNN or traditional quantum circuits, or even basics of quantum computing. What's a quantum gate?
Tanshu, I have a more general question as a non-computer scientist in the room.
Sure.
What was the biggest lesson that you learned here at ASDRP outside of the technical skills?
I think one thing would be kind of so when you research, when you learn something from your research, you have to document it. So we do have this like big giant sort of running project notebook where we dump out all our issues or learnings or everything. So I think it's very important because it's not just about you learning, like you can understand or I can understand whatever I'm reading, but to really like explain someone whatever I'm reading or understanding, that kind of takes my understanding to a next level. And it also helps the incoming people or the people who have not done the same things or read the same things as you did. Basically, it's kind of like crowdsourcing your knowledge. So you have 6 different people, they do different kinds of research. Now, if they all educate themselves and educate others, then all the 6 people know 6 different topics kind of thing. So, or different algorithms, depends what you're researching. But I think the main thing would be really understanding and then being able to explain and documenting that explanation in a common shared place.
Good. I think that's a lesson that a lot of people have to learn in research. So good on
Yeah,
you.
I think I'm going to give extra credit to Dipanshu in addition to his research, because this is what I'm like, that's my personal experience. When I was at UCSF over the holiday, the entire campus was closed, but I stepped in and wrote 300 line of code, and I was very excited to show it to my PI when it was detecting the functional connectivity of the brain. And when he looked at the code and he said, it's a trash. And it hurt me. It hurt me so much. And I said, did you see the result? Like, I don't understand. There's no documentation in that. There is nothing has been added as a human understandable word. It's a trash. So that was the time that it hurt me. It hurt me, but I understand as a researcher, if people cannot understand what you have done, basically your work is not valuable. So just a bunch of code in just getting your work done is not something that research is about. It's also about documenting, making sure that when people with no idea coming, they can trace your work and appreciate what you have done. So I'm just giving you extra credit. Good job.
Good job.
Thank you.
Yes. So let's see. I think we have one of my research students up to bat next. So I'll give a brief introduction to Shloka.
I think I got one quick question. I'll answer it.
Please.
That's okay. So, So the question was, what are 3 potential opportunities with QCNN compared to its predecessors? A bit vague, but I'm gonna try. I guess like there are different applications for QCNN, like speech recognition, like CNN by itself can be used for like video processing, which is like 3-dimensional, or it could be used for time series forecasting, which is 1-dimensional, or image recognition, which is 2-dimensional. But so QCNN ideally can be very good for video processing, where there's lots of data, and then maybe some QCNN for I don't know, like stock market prediction or any other time series sort of thing, like predicting any trends, stuff like that could be possible avenues of exploration. Yeah, so in general, yes, QCNN has a lot of, it can potentially adopt the same applications of CNN, and it can also solve, like, as I said before in the presentation, quantum mechanical problems that quantum computers really have an edge to compared to classical computers, like, I don't know, modeling the universe or predicting how a black hole would behave and under different conditions, things like that. Those are all possible applications. Yeah. Hope that answers your question.
So Shloka joined the group at Amador Valley. She joined this group when she was a freshman. She actually started her career at ASDRP working in a psychology group. And then she decided she wanted to do real science and moved into Soumya Suresh's group, where she worked on something related to plants and DNA and genetics and things like that. And after a semester in Soumya Suresh's group, joined my group, where I think she's been wondering what she got herself into for the last several years. And it's been a tremendous pleasure to have Shloka as one of my research students. You can always count on Shloka to be always very positive, always very excited to get work done, and always really excited about good results. So without further ado, Shloka, take it away. So the present button is how you present. On the right side, it says present.
Okay. Okay. Thanks for the introduction. So as I said, I'm Shloka, and I'll be presenting about the rational design and development of macrocyclic lactones with in vivo motility studies on nematode models and the scale-up synthesis of ribbostigmine analogs. So first, I'll start.
Hold on, sorry to interrupt. I don't think you're presenting your slides.
Really?
Yeah, it's just showing your whole screen. So,
Okay.
yes, you got that. Her connection. I think she got disconnected, so we'll wait a second for her to come back on. Let me Slack her real quick. Okay, she's having technical difficulties. Just wait a few minutes, a minute or so. So while we're waiting, I'm a little bit disappointed to see that there's only 140 some people here. This is mandatory for everybody to be part of. So if you all can do me a favor and tell your fellow researchers that attendance at Colloquium is mandatory. Yes. I think she's still having technical difficulties. Okay, I think her Zoom crashed, so she's going to try to rejoin. Give it like a minute or so. So, ladies and gents, we will take attendance at the very end for people who decide to show up. And if you are not here, if your friends are not here, please let them know that they will be getting an email from me if they are not here. Okay, I think she's back. Shloka, is everything working now?
Yeah.
There you go. All right, you got this. So let's see if it works better this time. There we go. So you gotta hit present. Cool, now we can see it. Take it away, Shloka.
Okay, so yeah, I'll be talking about the rational design and development of macrocyclic lactones with in vivo motility studies on nematode models and scalable synthesis of ribostigmine and its analogs. Okay, so here's a brief outline. So I'll first be talking about the overview of small molecules in neuroscience, and then the aggregation of the amyloid beta 42 peptide in Alzheimer's disease, and then just a few projects I've been working on, such as ribostigmine, or I guess projects surrounding ribostigmine, Thorium Actin, Compositing, and then I'll just end it with the conclusion and some acknowledgements. Okay, so as many of you may know, this is a neuron, and the various parts include the dendrites, axon, cell body, and the way it works is that the dendrites are the receptor proteins in the membrane that bind to the neurotransmitter to be activated. And then once activated, they initiate action potential, which is propagated through ion channels and terminated at the axon, which results in the release of more neurotransmitters, which is a pretty great target for drug development. So small molecules to alter neuron function by either engaging a receptor on the dendrite, blocking ion channels, or some that inhibit the uptake or degradation or release of neurotransmitters at the synapse. Okay, so my presentation kind of goes over four main molecules, well, main subtopics. So the first being polyphenols and its role in Alzheimer's disease, and then ribostigmine and its role in dementia, Thorium Actin as an anti-parasitic drug, and then vinpocetine, which is some relatively new work, but it relates to Parkinson's disease and stuff too. Okay, so the aggregation of the amyloid beta 42 or AB42 peptide in neurons causes the formation of amyloid plaques, which are linked to the development and progression of Alzheimer's disease. Increased levels of AB42 plaques have been found in early stages of Alzheimer's disease pathogenesis, resulting in neurotoxicity, the induction of oxidative stress, and eventually cell death of neurons. The inhibition of the amyloid protein aggregation has been investigated as a potential therapeutic strategy in the treatment and prevention of Alzheimer's. So this brings me to the first project, polyphenols. So polyphenols are a natural product small molecule produced in plants with various bioactive properties and have been previously demonstrated in inhibiting the aggregation of the AB42 peptide. We performed a systematic structure-activity relationship, or SAR, screen of a targeted library of natural product polyphenols for binding affinity to monomers and oligomers of the AB42 peptide by molecular docking simulations. Additionally, we performed an ex vivo thioflavin T aggregation assay, which would further be to justify our results from the SAR. So this would be our thioflavin T assay. And then that's just like the graph that we got. Results. So our results from the SAR indicate that the top three performing polyphenols were tannic acid, naringin, and rutin. And after analyzing their structure and performing MD simulations, which identifies which polyphenol destabilizes the AB42 oligomer, we noticed that glycosylated polyphenols have higher binding affinity and do better in stabilizing the monomer, possibly due to availability of hydrogen bond donors and acceptors of the compound. The high aggregation of the polyphenols of the AB42 peptide indicate potential therapeutics leads for Alzheimer's disease. Just more results. Okay, and then this is just some structures of the three best performing compounds. Okay, and then another method utilizes Tango, which is a software designed to predict the aggregation of the AB42 peptide along each amino acid. Our results indicate that amino acids 9 to 15 exhibit high aggregation, which provides further insight into what amino acids we should be targeting. So MD simulations were also performed using GROMACS to find which polyphenol best destabilizes the AB42 oligomer. Yeah, so this is just a whole bunch of our polyphenols, so EGCG, rutin, and origin. And then RMSD calculations were also conducted to find similarities between various atomic positions of the polyphenols. And RMSD basically shows how much the atom of AB42 changes over time, which can explain whether polyphenols destabilize or stabilize the oligomer. So from protein sequence-based RMSDs, the AB42 oligomer seems to have the most fluctuations in RMSD when simulated with EGCG specifically. And these polyphenols have high peaks in RMSD at specific amino acids, which show that they're most likely to be targeted by specific polyphenols. Okay, so now we'll be moving on to another group I've been working on for a while. So in this subgroup, we work on the compound ribbostigmine, which is a synthetic derivative of its natural occurring counterpart, bisostigmine. So ribbostigmine is an FDA-approved cognition-enhancing drug used commonly for the treatment of dementia and Alzheimer's. And as many of you know, Alzheimer's is very common, especially in old age. And according to the CDC, as of 2020, there's an estimated amount of 5.8 million Americans who have Alzheimer's, which makes our work pretty significant. So ribbostigmine irreversibly acylates the acetylcholine esterase enzyme, which is found in the synapse of a neuron. The synthesis of ribbostigmine can be potentially used to treat people with neurodegenerative diseases such as dementia, schizophrenia, and others. So in order to successfully synthesize ribbostigmine, we experimented using a variety of conditions, such as the ones reflected in this table. Our most efficient method was using DCM along with pyridine as a base and DMAP as a reaction catalyst. The experiment run with these reagents yielded a little over 30% products. Okay, so our synthetic efforts involve the synthesis of ribbostigmine and its analogs shown on this slide. To rationalize binding affinity, we ran docking studies on the 6 analogs in the acetylcholine esterase enzyme. Through DFT and docking, we predicted the binding affinity of each analog to the enzyme, and the lower the delta G, the more likely the compound is to have biological activity. As we see from our results, the des-methyl analog seems to have the lowest binding affinity, thus making it the most biologically interesting. So under some future directions, we'll be looking into performing a colorimetric acid in order to investigate the chemical activity of acetylcholine esterase. Essentially, the way it works is that the reactions lead to a change of color due to the enzymatic or chemical interaction. And in this case, it would be the enzymatic activity of the acetylcholine esterase enzyme. We'll be quantifying our results from this assay via microplate reader. Okay, and then sometimes we want to inhibit the neuron, which is what we focus on in studies surrounding ivermectins. People have found that ivermectin has been used to treat river blindness, which is caused by parasitic nematodes. And in such cases, we want to stop nematode neurons from functioning properly. Thorium Actin is a natural product derived from a soil organism. Belonging to a class of ivermectins, Thorium Actin is used as a veterinary drug to treat parasitic worms in cattle and swine. By upregulating chloride intake through activating glutamate-gated ion channels in parasitic nematodes, ivermectins induce paralysis and eventually death. In this study, we focus on Thorium Actin as a synthetic target. The removal of this ivermectin disaccharide yields an aglycone, which will then be utilized to create a variety of analogs in an attempt to increase anti-parasitic activity. Our goal is to achieve both sugar removal and oxidation to create a library of new Thorium Actin analogs. Thorium Actin has 2 methoxy sugar groups, and the deglycosylation or removing of sugars allows potentially interesting biological activity. Oxidizing Thorium Actin changes the alcohol group into ketone, which affects the compound cytotoxicity. There are 2 sites of oxidation on Thorium Actin that we can test, C5 and C13, and this can be achieved through multi-step reactions to get the final products. Using our compounds that were mentioned on the previous slide, we're going to test their biological activity on nematodes, specifically vinegar eels and C. elegans. Before that, however, we wanted to perform a few preliminary assays using abamectin, ivermectin, and doramectin to assess how one compound over the other could be seen as better or worse in their biological activity. So we tested this through performing motility assays to find how the movement of nematodes after the compound was administered was affected. So we'll also be using a script to be able to quantify the movement of vinegar eels and C. elegans. Here's a video of what we've got so far. So that's the vinegar eels under the fluorescent microscope. And then we administer the compound around 1 minute 30 seconds, and then we let it run for complete 10 minutes to see how many, like, I guess, die. Okay, so now I'll talk about the various steps taken in the computation side of our project. So first we start off with conformance searches, which allow us to gain insight into the structure and rotation of our compounds, and later serve as ligands in our docking studies. This is typically done using search algorithms with common force fields. The idea behind this is that large molecules such as Thorium Actin can adopt a large number of possible three-dimensional shapes, just like a hand can take different gestures, like a fist, a high five, stuff like that. And then using those compounds, we perform DFT, or density functional theory, which uses a quantum mechanical model to rigorously adjust three-dimensional structures to identify the most thermodynamically stable structure. The next step is homology modeling, in which we substitute amino acids in order to essentially predict the protein of interest. In doramectin, for example, the target protein, or the glutamate-gated chloride channel receptor in vinegar eels, has not been reported. So in order to proceed with docking studies, we have to predict the structure ourselves. So once we have the ligands from DFT and the protein from homology modeling, we perform docking in order to determine how our compounds bind to the protein using a software called AutoDock-Bina. And this process is fairly simple and quick and provides valuable insight into how our compounds bind to the protein. However, computational studies do not guarantee that one compound will work better than the other, which is why biological assays are really crucial to understanding how our compounds demonstrate their bioactivity. Okay, and then lastly, sometimes we want to target proteins inside the neuron. So this is our work regarding Compositing, which is a drug sold within the United States as a supplement to improve brain function, as it's been shown to increase blood flow to the brain, and it's been studied in the treatment of Alzheimer's. This is a pretty new subgroup, so we're still figuring out what synthetic efforts we want to pursue, but here is just a general schematic of what we're thinking. And then, so to wrap it up, a lot of people think chemistry is like mixing just random things together, seeing what happens, and just watching the reaction happen, which is kind of what we do. But then we also make sure we target specific human diseases, such as Alzheimer's that I've mentioned, and make molecules that really have an impact on everything. So in doramectin, our selective oxidation studies could be really interesting, and then quantification of vinegar eel movement using a script. And then in Ribbostigmine, just the complete synthesis of our Ribbostigmine analogs and our work in Compositing would also be very biologically interesting. Okay, so thank you to my advisor, Edward, for his support and guidance throughout my research experience here at ASDRP. I'd also like to thank the lab technicians for ensuring our safety every day we spend in lab. And lastly, I'd like to thank ASDRP as a whole and all of its corporate sponsors for supporting and funding our research. And of course, all the people I work with in Ed's group and even outside of Ed's group. So thank you, everyone. And I'd be happy to answer any questions.
Question. Great job, Shloka. I thought that was awesome. So there's the attendance sheet is in the polls. Anybody have questions for Shloka? So make sure you fill out the attendance so you don't get a nasty email from me tonight. Everybody who is not here will be getting a nasty email. Okay, Sanhita has a question.
I just want to ask, what do you think was the most like difficult or frustrating part of your work so far?
The most difficult part would be like trying reactions and just not having it work. And just like with our working ribostigmine, it's been like pretty frustrating sometimes since we've been doing this for like a while and we just like recently got ribostigmine, which is really great. But then along the way, it did kind of like, it was a struggle.
Got it.
All right, other questions? So I commend Shloka and her coworkers for going through a lot of pretty hard work. Synthetic chemistry is very challenging, and
CSCN, right?
some people like to announce which department they're part of. So great job to Shloka and her coworkers. Shloka is also a co-author on multiple manuscripts proceedings, and we're looking forward to many, many more. Okay. Oh, there's a question for you in the chat window, Shloka.
Okay.
Clinton Kunha and Michael Amati, who are both advisors here at ASDRP.
Okay, so for Clinton's question, so well, I guess if you're asking like, do all of them bind to like the same thing? They all bind to like different parts of the neuron, but then everything, like all of the work I've been like on does like bind directly to the neuron, but just different parts, I guess. And then also through like various literature and stuff, it has been shown that like ribostigmine is an acetylcholine esterase inhibitor. So yeah.
So, and then Michael has a question about where we got our ribostigmine from.
Oh, well, we made it. So here I can pull it up again. But after a long time and a lot of hours spent in lab, we were able to synthesize it. Oh, okay. And then Tavisha, my favorite instrument is the LCMS because it's great.
So we got more questions, one from Dr. McMahon. What is the precursor material for the synthesis?
Okay. Here, I'll just share my slides and then. Okay, so this is what our like synthetic route looks like, and then this is like the conditions that work the best for the reaction. If that's what you're asking.
So Larry, the starting phenol is commercially available. So we just buy that. We buy it in racemic mixture. Right now, this is a racemic product. You can get an enantiomer product with fractional crystallization. It's like a tartarate co-salt crystal or something like that. That's how industry prepares an enantiomer ribostigmine.